If you've watched my channel for a while, you know that I'm a fan of Dr. Roy L. Swank, who practiced at Oregon Health and Science University for many years and was the head of the neurology department before he began his own clinic focused on his low saturated fat diet. I was privileged to be his patient for a few years, and I followed the Swank diet for at least 5 years back in the early days of my MS, and I wonder if that didn't keep me from progressing to as great an extent as I might very well have. But I know that modern science looks at his work and they give you all kinds of reasons for why it's not valid, because he didn't have controls, he didn't have a double-blind experimental group, there were no MRIs back then, blah, 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 on and on. So even though many people still follow it, and even though there are modified versions of it that are promoted by other very well-known doctors, he's sort of fallen out of favor with many. But given the state of scientific knowledge at the time, I think he was pretty advanced. And new research shows he might actually have been more right than he even knew. Well, hello, and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam, and I've been living with MS for almost 40 years. I've made several videos over the years about Dr. Swank and his low-fat diet, which I would say is a healthy diet regardless, because he also talked even way back then about not having a lot of junk food in your diet. He didn't know everything we know about nutrition. There was no such thing as a double-blind, placebo-controlled clinical trial. There were no MRIs. Technology and science in general were a lot less advanced when he started his diet in the early 1950s. But he followed people for decades. And he saw some excellent results from the people who adhered to his diet, and he saw that the people who did not adhere tended to have more progression. Now, he didn't really know why. He had some theories, but he didn't know all that there was to know. But recently there have been several articles published that got me thinking about Dr. Swank again. And I want to share those with you because the conclusions that have come out of this are quite interesting, and at first maybe even a little counterintuitive, but they do, I think, vindicate Dr. Swank, and I think that you will find those interesting and encouraging. Here's an article from News Medical Life Sciences, and it was published on November 6th of 2024. It's titled, Saturated Fats Accelerate Neurodegeneration in Multiple Sclerosis. Researchers identify enzymes ceramide synthase 5 and 6 as key players in how saturated fats worsen multiple sclerosis symptoms. And here's another pretty picture showing the neuroprotective effect of neuron-specific deletion of the C16 ceramide synthetic enzymes in an animal model of multiple sclerosis. It's very attractive, I'll give it that, even if I don't really understand exactly what they're talking about yet, but let's see. We'll keep reading and see what we can find out. They say that in a recent study published in Glia, researchers examined how the synthesis of sphingolipid ceramide C16 from palmitic acid impacted the severity of neurodegenerative diseases such as multiple sclerosis. And I'm assuming that palmitic acid is a saturated fat, which doesn't seem to make sense because I thought palm oil was a plant. But what do I know? We're still learning, so let's keep reading. It says that the study used murine models of neurodegenerative diseases to examine whether inhibiting specific enzymes involved in ceramide C16 production reduced disease severity. And here's some background. They say that sphingolipids are important components of the cell membrane, especially in the brain and the myelin sheath on neurons, and play crucial roles in cell signaling. However, high levels of specific sphingolipids, such as ceramides, have been observed in various neurodegenerative diseases, such as Parkinson's disease, Alzheimer's disease, and multiple sclerosis. Recent studies have found elevated levels of ceramide C16 associated with increased cognitive decline and worse disease outcomes in individuals with multiple sclerosis and Alzheimer's disease, especially among people with high body mass index or those who consume diets rich in saturated fats. Dr. Swank would be smiling. I told you so. Ceramide C16 is produced either de novo, using substrates such as palmitic acid, abundant in palm oil. Okay, I was right about that. Or through the breakdown of other sphingolipids. Okay, so I was right. Palmitic acid is from palm oil, but somehow the ceramide C16 is produced basically from the components that are in palm oil. Interesting. The synthesis of ceramide C16 from saturated fats relies on specific enzymes. It is influenced by diets containing processed foods, rich in fatty acids, which increase ceramide levels and worsen neurodegenerative conditions. No argument there. And then they talk about the study. In the present study, the researchers used murine models that were neuronal knockouts for the genes encoding the enzymes ceramide syntheses, CERS. The genes encoding the enzymes CERS5 and CERS6, which synthesize the ceramides C14 and C16, were knocked out using mice with floxed alleles, which are genes that have been modified to allow selective deletion. The mice were then induced with experimental autoimmune encephalomyelitis to create a murine model of multiple sclerosis, for which they were injected with a specific myelin peptide and an adjuvant, and pertussis toxin was administered to increase disease severity. And here you go once again. We take a moment to thank the mice, whose death hopefully will result in some good for all of humanity. Additionally, the researchers derived primary neuronal cultures from rat embryo hippocampi and the hippocampal and cortical tissue of mouse embryos. The rat neuronal cells were then exposed to hydrogen peroxide to simulate oxidative stress, while the mouse neuronal cells were exposed to palmitic acid and hydrogen peroxide to simulate metabolic and oxidative stress, respectively. Quantitative real-time polymerase chain reaction, or RT-qPCR, was then conducted to measure the levels of messenger ribonucleic acid, mRNA, in the neuron and spinal cord samples, which allowed the researchers to analyze gene expression. The mRNA was converted to complementary deoxyribonucleic acid, or cDNA, through reverse transcription, after which the cDNA was amplified and quantified to assess gene expression levels. I think that you're seeing here that the audience for this particular article is probably a lot more technically savvy than most of us are, so I apologize for all of the terminology, but at least you're getting an idea of what they're doing to simulate the conditions that might be going on in the human body and finding out whether certain variables have a greater effect on multiple sclerosis. And they say that the study also included in situ hybridization experiments. So in situ means in place. So in this case, inside a mouse body, where the mouse brains were sectioned and processed to detect specific gene transcripts of CERS5 and CERS6 genes. The base scope assay, using labeled probes that bind specifically to target mRNA, was performed to detect the ceramide synthase gene transcripts. The brain tissue sections were also stained with antibodies specific for oligodendrocytes, and as you remember, the oligodendrocytes are the parts of our cells that make myelin. Neurons and astrocytes to identify the cell types. The labeled sections were then visualized using confocal microscopy, which allowed the researchers to observe the mRNA expression in different cell types. And here were the results. The results reported that lowering the production of ceramide C16 in the neurons offered neuroprotection in the murine multiple sclerosis model with experimental autoimmune encephalomyelitis. Specifically, deleting the CERS6 enzyme, which produces ceramide C16, in the excitatory neurons resulted in reduced disease severity and delayed onset of the disease, but only when the mouse was fed regular chow. When the mice were fed with a high-fat diet, the neuroprotective effect of CERS6 deletion was lost, which the researchers proposed was due to the increased activity of the CERS5 enzyme, which can also produce ceramide C16. Furthermore, when both CERS5 and CERS6 were deleted, the disease severity was lower even when the mice were fed the high-fat diet enriched with palmitic acid. This supported the finding that inhibiting the synthesis of ceramide C16 could reduce the severity of experimental autoimmune encephalomyelitis and multiple sclerosis. The study also found that ceramide C16 accumulation in the neurons affected mitochondrial function. When the neurons lacking the enzymes that produce ceramide C16 were exposed to palmitic acid, their mitochondria were more resilient, indicating that ceramide C16 accumulation compromised mitochondrial integrity in multiple sclerosis patients. And here are the conclusions. To summarize, the study found that limiting the synthesis of ceramide C16 in the neurons by targeting the enzymes CERS5 and CERS6 could be a potential therapeutic strategy to reduce disease severity and offer neuroprotective effects in multiple sclerosis patients. Dietary modifications consisting of lower fatty acid consumption, especially of palmitic acid sources such as palm oil, could also help in mitigating disease severity in demyelinating disorders. Well, this is an interesting finding because what it says to me is that avoiding the normal bad actors when it comes to saturated fats, and I think we all know what those are, butter and certain meats and dairy products of various kinds, is not enough because palm oil is not something that I normally would have associated with high levels of saturated fat. But if the building blocks of palm oil could be reconfigured in the body to create something that's basically saturated fat, we really do have to be careful what we eat. I know a lot of nutritionists talk about the dangers of seed oils, and that would include palm oil. And this definitely underscores that they're onto something for sure. And a few days earlier, on November 1st, this article appeared in the same source we just looked at. It is called Research Identifies Crucial Links Between Dietary Choices and Progression of Multiple Sclerosis. They say that newly published research in the journal Glia has identified crucial links between dietary choices and the progression of multiple sclerosis. The study, led by Patrizia Casaccia, founding director of the Advanced Science Research Center at the City University of New York Graduate Center, Neuroscience Initiative, and Einstein Professor of Biology and Biochemistry at the CUNY Graduate Center, explored how enzymes called ceramide synthase 5 and 6 are responsible for the toxic effect of a palm oil-rich diet on neurons in the central nervous system, which causes a subsequent increase in the severity of MS symptoms. So this looks like it's reporting on the same research, but let's hope maybe in a way that's a little bit more easy to understand. They have a paragraph that talks about MS as an inflammatory autoimmune disease. I don't think we need to read through that because we've heard this many, many times. But you can see this last sentence where they say, in their study, researchers explored potential mechanisms by which a diet rich in palm oil may hijack neuronal health. They talk about neuroprotection from palm oil-induced toxicity. Using the experimental autoimmune encephalomyelitis model, or the EAE model, of inflammatory demyelination, the research team found that diets high in palm oil led to a more severe disease course in mice. Yeah, whenever you see EAE, think mice, because that's the MS-like disease that scientists are able to induce in mice. And then they can study the effect of various things on the mice that they have been artificially given this EAE. They say here in the quote, "We reasoned that inside neuronal cells, palm oil is converted into a toxic substance called C16 ceramide by specific enzymes called CERS5 and CERS6. The ceramide is responsible for inflicting mitochondrial damage, which deprives neurons of the energy they need to counteract inflammation in the brain. We therefore asked whether inactivation of these enzymes would confer neural protection." And this quote is from the lead investigator, Patrizia Casaccia. They say that the researchers found that when they genetically deleted the enzymes CERS6 and CERS5 in neurons, they could prevent neurodegeneration in the experimental model of MS. This held true even when mice were fed a diet rich in palmitic acid, said the paper's first author, Damien Marichal, a research associate with the Casaccia lab. This new information points to a specific metabolic pathway through which dietary fats can worsen MS symptoms. Isn't that fascinating? And I just wish Dr. Swank could see this research, because I would think he'd be quite interested. And they talk about the significance for MS patients and clinicians. The paper's findings have significant implications for individuals diagnosed with MS, as well as for clinicians treating patients and neuroscientists researching the disease. The work reinforces that lifestyle choices such as diet can have a profound impact on the course of the disease. The study's results build on previous concepts about careful dietary choices in managing the symptoms of MS. The findings also identify potential molecules that could help slow diet-induced symptom severity. Well, that's interesting too, because they're talking about symptoms here. And I'm not sure what they're talking about is sort of the underlying mechanisms and how to stop that, but it might reduce the severity of our symptoms, I think, because it's targeting inflammation, which is really the key culprit in a lot of our poor dietary choices. But let's continue on with what they have to say. "Our research provides a molecular explanation for how to protect neurons from the palm oil-dependent creation of molecules that harm them," said Casaccia. "We hope this information can empower patients to make informed dietary decisions that could positively impact the course of the disease while identifying strategies to counteract the effect of CERS5 and CERS6 in a neuron-specific fashion." And the research was funded by the National Institute of Neurological Disorders and Stroke of the National Institutes of Health. Well, armed with all this knowledge, what are we supposed to do? Science once again comes to the rescue, because there's an article in the same source we've been looking at that was published on November 4th of 2024. Scientists discover how the keto diet may help some people with multiple sclerosis. Right off the bat, you're thinking, wait a minute, I didn't think keto was as concerned about restricting saturated fat as it was with restricting carbohydrates. So let's read on and find out what they're talking about. Scientists have long suspected the keto diet might be able to calm an overactive immune system and help some people with diseases like multiple sclerosis. Now they have reason to believe it could be true. Scientists at UC San Francisco have discovered that the diet makes the gut and its microbes produce two factors that attenuated symptoms of MS in mice. If the study translates to humans, and of course this is a big if, it points toward a new way of treating MS and other autoimmune disorders with supplements. Wouldn't that be a nice change from pharmaceuticals? The keto diet severely restricts carbohydrate-rich foods like bread, pasta, fruit, and sugar, but allows unlimited fat consumption. Okay, so there's the counterintuitive aspect of this particular finding. Without carbohydrates to use as fuel, the body breaks down fat instead, producing compounds called ketone bodies. Ketone bodies provide energy for cells to burn and can also change the immune system. Working with a mouse model of MS, there's those little mice again, the researchers found that mice who produced more of a particular ketone body called beta-hydroxybutyrate had less severe disease. The additional beta-HB also prompted the gut bacterium Lactobacillus murinus to produce a metabolite called indole lactic acid, or ILA. This blocked the activation of T helper 17 immune cells, which are involved in MS and other autoimmune disorders. What was really exciting was finding that we could protect these mice from inflammatory disease just by putting them on a diet that we supplemented with these compounds. And that's Peter Turnbach, PhD of the Benioff Center for Microbiome Medicine. Earlier, Turnbach had shown that when secreted by the gut, beta-HB counteracts immune activation. This prompted a postdoctoral scholar who was then working in his lab, Margaret Alexander, PhD, to see if the compound could ease the symptoms of MS in mice. In the new study, which appears November 4th in Cell Reports, the team looked at how the ketone body-rich diet affected mice that were unable to procure beta-HB in their intestines and found that their inflammation was more severe. But when the researchers supplemented their diets with beta-HB, the mice got better. To find out how beta-HB affects the gut microbiome, the team isolated bacteria from the guts of 3 groups of mice that were fed either the keto diet, a high-fat diet, or the beta-HB supplemented high-fat diet. Then they screened the metabolic products of each group's distinct microbes in an immune assay and determined that the positive effects of the diet were coming from a member of the Lactobacillus genus, L. murinus. 2 other techniques, genome sequencing and mass spectrometry, confirmed that the L. murinus that they found produced indole lactic acid, which is known to affect the immune system. Finally, the researchers treated the MS mice with either ILA or L. murinus, and their symptoms improved. Turnbach cautioned that the supplement approach still needs to be tested in people with autoimmune disorders. So just because it works in mice, don't go thinking it's going to work in you. The big question now is how much of this will translate into actual patients, he said. But I think these results provide hope for the development of a more tolerable alternative to helping those people than asking them to stick to a challenging, restrictive diet. Well, I admit I found this kind of perplexing because somehow this idea of saturated fat being bad but the keto diet being good, I was having trouble reconciling that. And I'm not really sure I've come to the total bottom line conclusion here. But this portion of an article from Multiple Sclerosis News Today that was published on November 8th of 2024, there's a section in here that I think is quite helpful. The article is called Keto Diet Modifies Gut Bacteria in Mice, Protecting Against MS. Ketogenic Diet Found to Lessen Severity of Multiple Sclerosis in Mouse Models. And this article goes over basically the same research we looked at in the other source, but this section I think was a little more understandable and helpful. So I'm just going to read this part. Molecules made by gut bacteria key to beneficial effects of keto diet. Diet can have profound effects on health for all people, but especially those with autoimmune diseases like MS or other conditions. A ketogenic or keto diet involves minimizing the intake of carbohydrates, which forces the body to burn fat for energy. The diet gets its name because when the body is using fats as its main source of energy, it makes molecules called ketone bodies. There isn't any one diet recommended for people with MS, although the keto diet is one dietary strategy that is sometimes explored by people with the disease. Some small studies have indicated that this eating plan may help to ease fatigue and depression, and experimental evidence also suggests that a keto diet can lessen disease severity in animal models of MS. However, it hasn't been clear why exactly this type of diet shows benefits in MS models. In this study, the researchers wanted to determine whether the impact of a keto diet in MS models is dependent on the gut microbiome, the billions of bacteria and other microorganisms that live in the intestinal tract. In their initial experiments, the researchers induced experimental autoimmune encephalomyelitis, or EAE, an MS-like disease, in mice, then fed the animals either a high-fat diet or a ketogenic one. In line with prior research, mice given the ketogenic diet had much less severe disease. The researchers then repeated the experiment using germ-free mice that were engineered to completely lack a gut microbiome. Contrasting with the original findings, disease severity was comparable in germ-free mice fed a ketogenic or high-fat diet, illustrating that the impact of the ketogenic diet in this model is dependent on gut bacteria. Okay, that's a finding, I guess. Most of us are not going to have to worry about having a gut that's germ-free, but I understand what they're trying to do here. When mice are fed a ketogenic diet, a ketone body called beta-hydroxybutyrate, or beta-HB, is significantly increased in circulation relative to the high-fat diet. This molecule is known to modulate the activity of inflammatory immune cells, so the researchers speculated that reduced production of beta-HB in germ-free mice might explain why these mice didn't benefit from the keto diet. So, in other words, it's the beta-hydroxybutyrate that makes the difference. And if you don't have that in your gut, or you don't have it in sufficient quantities, you are more prone to suffer from the inflammation that drives the symptoms of MS. I think I got that right. And that would mean that the ketogenic diet, which metabolically produces more of this compound, is going to be beneficial to those of us who have MS, and probably beneficial to everybody else as well. At least that's the theory. Sounds sort of plausible. Supporting this idea, the researchers found that supplementing the mice's high-fat diet with beta-hydroxybutyrate led to less severe disease, similar to that seen with the keto diet. We discovered that oral delivery of a beta-HB can mimic the protective effects of a ketogenic diet, the researchers wrote. The translational implications from animals to humans are profound, as ketogenic diets are difficult to maintain and can have negative side effects. Well, I'm glad that they did make a point of that. I think sticking to any kind of a diet that restricts what you can eat restricts a lot more than just our choices that we make at home, but it restricts our ability to go to friends' houses, or to go out to eat, or to go to your church potluck, or anything else for that matter. And if they've isolated a compound that, if we could supplement our diets with that, would take the place of having to have a strict keto diet, maybe that would be a beneficial thing for us to do. Because once again, you have to reduce the inflammation, because that drives a lot of the negative side effects and symptoms of multiple sclerosis. Well, I'll go for that. Well, Dr. Swank was not a big believer in having a control in an experiment if he knew already, or if he strongly suspected that the control group was going to suffer because the intervention was so helpful. And that's exactly what happened with the low saturated fat diet that he promoted. It wasn't like he didn't know there were such things as controls in experiments, because that's been around for a long time. But he thought it was unethical to put someone on a regular diet when he saw from the results of decades that people who avoid saturated fats did better. Maybe he wasn't ticking all the boxes for scientific rigor that we believe today is so important, But his heart was certainly in the right place, and as an ethical doctor, you can't beat him. Like I say, I'm very happy to be sharing this information with you because I think if we get to the bottom of this idea of this beta hydroxybutyrate as a compound that, if you don't have enough of it, causes problems, that might be a really important step in the direction of figuring out the link between what we eat or don't eat and how we feel and how our symptoms progress. Well, that's all I have for you today. Leave me a comment on this. Tell me if you've ever tried the Swank diet or if you would like to try it. But I'll see you again in my next video.